All right, everyone, we have some more interesting bug news today. This story I saw on BBC. X-ray reveals tiny half-billion-year-old creature. So what's cool about this is, this fossil was found in China, and it was absolutely tiny, and it was a fossilized larva, pre-insect, kind of generic arthropod larva. And originally, they weren't entirely sure what it was, but researchers quickly realized that there was something strange about this particular fossil because it was in such good condition. This is small enough to fit on the head of a pin, and once this fossil was taken back to the UK, they did a bunch of X-ray testing on this, X-ray scans of this 520 million year old fossil, and they got a bunch of internal structures that were largely intact. They're saying that this is because it was preserved in a high phosphorus environment, so phosphorus flooded the specimen and allowed a bunch of stuff to be seen, or a bunch of the soft tissue to be preserved. And what was cool, and I'll link this as well. This comes out of the journal Nature, and I'll link the original article as well because it is really interesting. But a lot of the soft structure cavities were preserved. So you can actually see kind of the pre-insect morphology and the evolutionary chain of this. For those of you who don't normally see my videos, I have a introduction to insect morphology course that I've been posting for the last about month or so, 6 weeks. I think I just finished part 20, which is on the brain and the nervous system of insects. But you can see a lot of these structures in the insect. So up here, these are the soft tissues of the brain. So this pink here is the frontal brain body. The teal is the central brain body here, and then this blue is the dorsal brain body. Let's clear that out a little bit. So those three tissues are visible there. Here in the front in orange, let me change this to orange. Here in orange on the front, you see the stomodium. So this is the sclerotized chitinous tissue of the esophagus, and the proctodium is slightly visible here. This is the rectum and the anus, which is also sclerotized and chitinous. And you can see that a little bit down here. I don't know why this copied in this way, but the stomodium is visible. In red along the dorsal surface, they were able to visualize the circulatory system here, which isn't super visible on this compound image, and that's just because a lot of other tissue is covering up the dorsal circulatory system, which would be running right here in red. What else do we have visible here? We have the paravisceral cavity, which is what you see here in all this teal. Let's do this in black. So all of this sort of teal tissue here, this is the paravisceral cavity. This is the tissue that would be surrounding all of the organs, which is still largely intact. A lot of the things that are intact are things which would have sclerotized sort of membranes around them, or structural membranes around them. And down here in purple, this would be the cavity around the nerve cord, the ventral nerve cord, extending from the brain. So these are things which you will still see in insects today, but which are highly developed, highly specialized in insects. And in this particular specimen, because it's so old and it predates all of our modern insects, you have these very generalized structures. You don't have the high specialization that you see in modern insects. This sort of ugly mustard yellow color up here is part of the digestive system. These are glands that would normally surround the digestive tract. The digestive tract itself, all that's left is the orange portions here. The entire central midgut of insects, let me clear this all up. There's so much going on here. The entire central midgut of insects, which would normally be between these two orange points, is soft tissue. It's not chitinous, it's not sclerotized, and so all of that likely rotted very, very quickly. Partially because it's soft and partially because it's already full of bacteria. And generally with insects, as soon as they die, that sort of tissue breaks down immediately. It's very thin and very fragile, so you're not going to see fossilized remnants of that. There is some here, up here, barely visible, are the precursors to modern insect compound eyes. This would be the sclerotized portions of the eye, which are still preserved. What's interesting about this is that the brain in the actual breakdown of these images, the brain extends backwards quite a bit. Because this particular specimen predates modern insects, the compound head hasn't been formed yet. And I go through this quite a bit in my morphology series. The head is actually composed of between 5 and 8 separate body segments that have fused together, and all of the ganglia have fused together. That hasn't completely occurred yet, so you have this gangliar chain from the brain going backwards. In modern insects, all of these segments that you would see in this blue would likely be fused into a contiguous head. So this would be where you get the mandibles, the maxillae, the labium, the compound eyes, the antennae, and things like that, which would normally in the primitive arthropod, all of those structures would just be legs, which would then be fused onto the head. So that hasn't occurred yet. So it's really interesting if you're interested in kind of paleontology and ancient insects and stuff like that. But a lot, you can see just from this X-ray scan, a lot of the soft structure is intact. So I'll link both this article from BBC and the original Nature article for your reading pleasure if you're interested. And I'll talk to you guys later. Thanks.